Blood clotting is an important protective function. It ensures that when you injure yourself, you don't bleed for too long. However, if the blood clots at the wrong time, for instance within a blood vessel, this can be catastrophic. In fact, this is known as thrombosis, and thrombosis is ultimately what leads to heart attacks and strokes. This happens because it stops the flow of normal oxygenated blood to your tissues, such as your heart or your brain. Within the blood are tiny blood cells known as platelets, and their job is to recognise tissue injury and when they see tissue injury to respond and cause the blood to clot. Now, it's been recognised for a number of years now that drugs that are able to suppress the function of these cells to make them respond less strongly, if you like, are able to prevent thrombosis. In order to develop better drugs, safer drugs and more effective drugs that prevent thrombosis, it's important that we know as much as we can about how these cells work. To do this, we think of the platelet, actually like many other cells, like a little machine. It's got lots and lots of tiny components within it. We can't see them, so we have to develop sophisticated techniques that enable us to study how these different components of the machine work together to enable the cell to respond so rapidly when it encounters tissue damage. In recent years, we've identified a number of important molecules on platelets, particularly molecules that enable them to see tissue injury or molecules that are exposed when the circulation becomes damaged. Recently, we've identified some hormone-like molecules that are released by platelets when they become activated when they encounter injury, and these send out a message to other platelets to tell them to become activated and to recruit them into a thrombus or a clot that stems the flow of blood. We've also identified the molecules on the cell surface that enable platelets to respond to these hormone-like molecules that they release during activation, and drugs already exist that are able to block these receptors. So ongoing work is trying to establish whether this could be a new approach to prevent thrombosis using molecules drug-like molecules that we already know are safe to use. The big challenge to researchers working in this area is that effectively what we're doing is suppressing the function of a normal protective role, and we need to make sure that we're able to suppress platelet function to the point where it prevents thrombosis, but it doesn't prevent normal blood clotting. It's also probable that this will be combinations of approaches. There's no simple solution to this particular problem, and in different individuals they may be given different combinations of drugs, but this would also be combined with other effects such as nutritional or changes in the diet, maybe exercise, other effects that are known to have substantial roles in the development or ongoing development of cardiovascular diseases.